Magnetoencephalography is a functional neuroimaging technique for mapping brain activity by recording magnetic fields produced by electrical currents occurring naturally in the brain, using very sensitive magnetometers. Arrays of SQUIDs are currently the most common magnetometer, while the SERF is being investigated for future machines. Applications of MEG include basic research into perceptual and cognitive brain processes, localizing regions affected by pathology before surgical removal, determining the function of various parts of the brain, and neurofeedback. This can be applied in a clinical setting to find locations of abnormalities as well as in an experimental setting to simply measure brain activity. History of MEG. MEG signals were first measured by University of Illinois physicist David Cohen in 1968, before the availability of the SQUID, using a copper induction coil as the detector. To reduce the magnetic background noise, the measurements were made in a magnetically shielded room. The coil detector was barely sensitive enough, resulting in poor, noisy MEG measurements that were difficult to use. Later, Cohen built a better shielded room at MIT, and used one of the first SQUID detectors, just developed by James E. Zimmerman, a researcher at Ford Motor Company, to again measure MEG signals. This time the signals were almost as clear as those of EEG. This stimulated the interest of physicists who had been looking for uses of SQUIDs. Subsequent to this, various types of spontaneous and evoked MEGs began to be measured. At first, a single SQUID detector was used to successively measure the magnetic field at a number of points around the subjector unregistered trademark S head. This was cumbersome, and, in the 1980s, MEG manufacturers began to arrange multiple sensors into arrays to cover a larger area of the head. Present-day MEG arrays are set in helmet-shaped duo that typically contain 300 sensors, covering most of the head. In this way, MEGs of a subject or patient can now be accumulated rapidly and efficiently. Recently it has been demonstrated that MEG could work with a chip-scale atomic magnetometer. 1. The basis of the MEG signal, synchronized neuronal currents induce weak magnetic fields. At 10 femtotesla for cortical activity and 103 feet for the human alpha rhythm, the brain's magnetic field is considerably smaller than the ambient magnetic noise in an urban environment, which is on the order of 108 feet or 0.1 micron T. The essential problem of biomagnetism is, thus, the weakness of the signal relative to the sensitivity of the detectors, and to the competing environmental noise. The MEG signals derive from the net effect of ionic currents flowing in the dendrites of neurons during synaptic transmission. In accordance with Maxwell's equations, any electrical current will produce an orthogonally oriented magnetic field. It is this field that is measured. The net currents can be thought of as current dipoles, that is currents with a position, orientation, and magnitude, but no spatial extent. According to the right-hand rule, a current dipole gives rise to a magnetic field that flows around the axis of its vector component. To generate a signal that is detectable, approximately 50,000 active neurons are needed. Since current dipoles must have similar orientations to generate magnetic fields that reinforce each other, it is often a layer of pyramidal cells, which are situated perpendicular to the cortical surface, that gives rise to measurable magnetic fields. Bundles of these neurons that are orientated tangentially to the scalp surface project measurable portions of their magnetic fields outside of the head, and these bundles are typically located in the sulci. Researchers are experimenting with various signal processing methods in the search for methods that detect deep brain signal, but no clinically useful method is currently available. It is worth noting that action potentials do not usually produce an observable field, mainly because the currents associated with action potentials flow in opposite directions and the magnetic fields cancel out. However, action fields have been measured from peripheral nerves. Magnetic shielding. Since the magnetic signals emitted by the brain are on the order of a few femtoteslas, shielding from external magnetic signals, including the Earth's magnetic field, is necessary. Appropriate magnetic shielding can be obtained by constructing rooms made of aluminium and mu metal for reducing high-frequency and low-frequency noise, respectively. Magnetically shielded room. A magnetically shielded room model consists of 3 nested main layers. Each of these layers is made of a pure aluminium layer, plus a high permeability ferromagnetic layer, similar in composition to molybdenum permalloy. The ferromagnetic layer is supplied as 1 ohm sheets, while the innermost layer is composed of 4 sheets in close contact, and the outer 2 layers are composed of 3 sheets each. Magnetic continuity is maintained by overlay strips. Insulating washers are used in the screw assemblies to ensure that each main layer is electrically isolated. This helps eliminate radio frequency radiation, which would degrade SQUID performance. Electrical continuity of the aluminium is also maintained by aluminium overlay strips to ensure AC eddy current shielding, which is important at frequencies greater than 1 Hz. The junctions of the inner layer are often electroplated with silver or gold to improve conductivity of the aluminium layers. Active shielding system. Active systems are designed for 3-dimensional noise cancellation. To implement an active system, low noise flux gate magnetometers are mounted at the center of each surface and oriented orthogonally to it. This negatively feeds a DC amplifier through a low-pass network with a slow fall-off to minimize positive feedback and oscillation. Built into the system are shaking and degaussing wires. Shaking wires increase the magnetic permeability, while the permanent degaussing wires are applied to all surfaces of the inner main layer to degauss the surfaces. Moreover, noise cancellation algorithms can reduce both low-frequency and high-frequency noise. Modern systems have a noise floor of around 2 foot per Hertz, 0.5 above 1 Hz. Source localization, the inverse problem. The challenge posed by MEG is to determine the location of electric activity within the brain from the induced magnetic fields outside the head. Problems such as this, where model parameters have to be estimated from measured data, are referred to as inverse problems, are known and the data is to be estimated. The primary difficulty is that the inverse problem does not have a unique solution, and the problem of defining the best solution is itself the subject of intensive research. Possible solutions can be derived using models involving prior knowledge of brain activity. The source models can be either overdetermined or underdetermined. An overdetermined model may consist of a few point-like sources, whose locations are then estimated from the data. Underdetermined models may be used in cases where many different distributed areas are activated. There are infinitely many possible current distributions explaining the measurement results, but the most likely is selected. Localization algorithms make use of given source and head models to find a likely location for an underlying focal field generator. Localization algorithms using overdetermined models operate by expectation maximization. The system is initialized with a first guess. A loop is started, in which a forward model is used to simulate the magnetic field that would result from the current guess. The guess is adjusted to reduce the discrepancy between the simulated field and the measured field. This process is iterated until convergence. The extent to which the constraint-free MEG inverse problem is ill-posed cannot be overemphasized. If one's goal is to estimate the current density within the human brain with say a 5 mm resolution, then it is well established that the vast majority of the information needed to perform a unique inversion must come not from the magnetic field measurement but rather from the constraints applied to the problem. Furthermore, even when a unique inversion is possible in the presence of such constraints, said inversion can be unstable. These conclusions are easily deduced from published works. Magnetic source imaging. The estimated source locations can be combined with magnetic resonance imaging images to create magnetic source images. The two sets of data are combined by measuring the location of a common set of fiducial points marked during MRI with lipid markers and marked during MEG with electrified coils of wire that give off magnetic fields. The locations of the fiducial points in each data set are then used to define a common coordinate system so that superimposing the functional MEG data onto the structural MRI data is possible. A criticism of the use of this technique in clinical practice is that it produces colored areas with definite boundaries superimposed upon an MRI scan. The untrained viewer may not realize that the colors do not represent a physiological certainty. Because of the relatively low spatial resolution of MEG, but rather a probability cloud derived from statistical processes. However, when the magnetic source image corroborates other data, it can be of clinical utility. Dipole model source localization. A widely accepted source modeling technique for MEG involves calculating a set of equivalent current dipoles, which assumes the underlying neuronal sources to be focal. This dipole fitting procedure is nonlinear and overdetermined, since the number of unknown dipole parameters is smaller than the number of MEG measurements. Automated multiple dipole model algorithms such as multiple signal classification and MSST modeling are applied to the analysis of MEG responses. The limitations of dipole models for characterizing neuronal responses are difficulties in localizing extended sources with ECDs, problems with accurately estimating the total number of dipoles in advance, and dependency on dipole location, especially depth in the brain. Distributed source models. Unlike multiple dipole modeling, distributed source models divide the source space into a grid containing a large number of dipoles. The inverse problem is to obtain the dipole moments for the grid nodes. As the number of unknown dipole moments is much greater than the number of MEG sensors, the inverse solution is highly underdetermined, so additional constraints are needed to reduce ambiguity of the solution. The primary advantage of this approach is that no prior specification of the source model is necessary. However, the resulting distributions may be difficult to interpret, because they only reflect a blurred image of the true neuronal source distribution. The matter is complicated by the fact that spatial resolution depends strongly on several parameters such as brain area, depth, orientation, number of sensors, etc. Independent component analysis. Independent component analysis is another signal processing solution that separates different signals that are statistically independent in time. It is primarily used to remove artifacts such as blinking, eye muscle movement, facial muscle artifacts, cardiac artifacts, etc. from MEG and EEG signals that may be contaminated with outside noise. However, ICA has poor resolution of highly correlated brain sources. MEG use in the field. In research, MEG's primary use is the measurement of time courses of activity. MEG can resolve events with a precision of 10 milliseconds or faster, while functional MRI, which depends on changes in blood flow, can at best resolve events with a precision of several hundred milliseconds. MEG also accurately pinpoints sources in primary auditory, somatosensory, and motor areas. For creating functional maps of human cortex during more complex cognitive tasks, MEG is most often combined with fMRI, as the methods complement each other. Neuronal and hemodynamic data do not necessarily agree, in spite of the tight relationship between local field potentials and blood oxygenation level-dependent signals. MEG and bold signals may originate from the same source. MEG is also being used to better localize responses in the brain. The openness of the MEG setup allows external auditory and visual stimuli to be easily introduced. Some movement by the subject is also possible as long as it does not jar the subjector unregistered trademark S head. The responses in the brain before, during, and after the introduction of such stimuli movement can then be mapped with greater spatial resolution than was previously possible with EEG. Psychologists are also taking advantage of MEG neuroimaging to better understand relationships between brain function and behavior. For example, a number of studies have been done comparing the MEG responses of patients with psychological troubles to control patients. There has been great success isolating unique responses in patients with schizophrenia, such as auditory gating deficits, to human voices. MEG is also being used to correlate standard psychological responses, such as the emotional dependence of language comprehension. Recent studies have reported successful classification of patients with multiple sclerosis, Alzheimer's disease, schizophrenia, Star-Paragraph-Grenz syndrome, chronic alcoholism, and facial pain. MEG can be used to distinguish these patients from healthy control subjects, suggesting a future role of MEG in diagnostics. Focal epilepsy. The clinical uses of MEG are in detecting and localizing pathological activity in patients with epilepsy, and in localizing eloquent cortex for surgical planning in patients with brain tumors or intractable epilepsy. The goal of epilepsy surgery is to remove the epileptogenic tissue while sparing healthy brain areas. Knowing the exact position of essential brain regions helps to avoid surgically induced neurological deficits. Direct cortical stimulation and somatosensory evoked potentials recorded on ECOG are considered the gold standard for localizing essential brain regions. These procedures can be performed either intraoperatively or from chronically indwelling subdural grid electrodes. Both are invasive. Non-invasive MEG localizations of the central sulcus obtained from somatosensory evoked magnetic fields show strong agreement with these invasive recordings. MEG studies assist in clarification of the functional organization of primary somatosensory cortex and to delineate the spatial extent of hand somatosensory cortex by stimulation of the individual digits. This agreement between invasive localization of cortical tissue and MEG recordings shows the effectiveness of MEG analysis and indicates that MEG may substitute invasive procedures in the future. Fetal MEG has been used to study cognitive processes such as vision, audition, and language processing in fetuses and newborns. Comparison with related techniques, MEG has been in development since the 1960s but has been greatly aided by recent advances in computing algorithms and hardware, and promises improved spatial resolution coupled with extremely high temporal resolution. Since the MEG signal is a direct measure of neuronal activity, its temporal resolution is comparable with that of intracranial electrodes. MEG complements other brain activity measurement techniques such as electroencephalography, positron emission tomography, and fMRI. Its strengths consist in independence of head geometry compared to EEG, non-invasiveness and use of no ionizing radiation, as opposed to PET. MEG versus EEG. Although EEG and MEG signals originate from the same neurophysiological processes, there are important differences. Magnetic fields are less distorted than electric fields by the skull and scalp, which results in a better spatial resolution of the MEG. Whereas scalp EEG is sensitive to both tangential and radial components of a current source in a spherical volume conductor, MEG detects only its tangential components. Scalp EEG can, therefore, detect activity both in the sulci and at the top of the cortical gyri, whereas MEG is most sensitive to activity originating in sulci. EEG is, therefore, sensitive to activity in more brain areas, but activity that is visible in MEG can also be localized with more accuracy. Scalp EEG is sensitive to extracellular volume currents produced by postsynaptic potentials. MEG detects intracellular currents associated primarily with these synaptic potentials because the field components generated by volume currents tend to cancel out in a spherical volume conductor. The decay of magnetic fields as a function of distance is more pronounced than for electric fields. Therefore, MEG is more sensitive to superficial cortical activity, which makes it useful for the study of neocortical epilepsy. Finally, MEG is reference-free, while scalp EEG relies on a reference that, when active, makes interpretation of the data difficult. See also, electroencephalography, electrophysiology, evoked field, auditory evoked field, functional neuroimaging, magnetocardiography, magnetogastrography, magnetomeography, magnetometer, magnetic source imaging, mu-metal, SQUID, direct brain interfaces, whole brain emulation, field trip. Further reading, Baylet S., Moshe J.C., Lee E.R.M. Electromagnetic Brain Mapping in IEEE Signal Processing Magazine, November 2001, 14-30. Cohen, D. Boston and the History of Biomagnetism. Neurology and Clinical Neurophysiology 2004. 31. Cohen, D. Halgren, E. Magnetoencephalography. In Encyclopedia of Neuroscience, Adelman G., Smith B., editors Elsevier, 1st, 2nd and 3rd editions. Ha currency Ma currency La currency M., Hari, R., Ilmoniemi, R., Kumutila, J. and Lau Nasma, O.V. Magnetoencephalography, a Eurotheory, Instrumentation, and Applications to Non-Invasive Studies of Signal Processing in the Human Brain and Reviews of Modern Physics 1993, 65, ppA 413 a €497, Hansen, Peter C., Krinchlbach, Morton Allen Solmerlin, Rita MEG, An Introduction to Methods. New York, Oxford University Press Incorporated. Murakami S., Okada Y. Contributions of Principal Neocortical Neurons to Magnetoencephalography and ELECTR, Encephalography Signals. J. Feisal, September 15, 2006. 575, pt 3, 925-36. Suk, J., Ribari, U., Capel, J. Yamamoto, T. and Linas, are anatomical localization revealed by MEG recordings of the human somatosensory system. EEG J78 185-196, 1991. Tanzaroguz 1, Numerical Modeling in Electro- and Magnetoencephalography, Ph.D. Thesis, Helsinki University of Technology, Finland. References